Let's now talk about the sodium-potassium pump. Sodium-potassium pump is going to be a protein that's found in the cytoplasmic membrane. So in the plasma membrane, which is going to be composed of a phospholipid bilayer, these are phospholipids, we're going to insert a protein in here. And for my sake, I'm going to draw it by using two vertical sides, and these are going to undergo a conformation change when ATP is attached to it. So right now, we're going to orient you. So here's our phospholipid bilayer. This is going to be inside the cell, and this is going to be outside the cell. So here's going to be cytoplasm from here on down, and here's going to be the extracellular space. Here's more, so this will be outside the cell, this will be inside, out, in, and then finally out, in. So we have four different phospholipid bilayers. Anything above the phospholipid bilayer is going to be outside the cell. Anything below the phospholipid bilayer is going to be inside the cell. So there's our orientation. We all know that sodium likes to be outside the cell, and we all know that potassium likes to be inside the cell. So this pump is going to put potassium inside the cell and sodium outside the cell. We're going to build a gradient. So we're going to go against the gradient. We know that sodium diffuses into the cell and potassium will diffuse outside of the cell. So what this pump is we're going to be building a sodium gradient outside the cell and a potassium gradient in. Just for clarification here, potassium is normally found in the cell. Sodium is found outside the cell. This is caused by this sodium-potassium pump because we have such a high potassium gradient outside that it will want to go into the cell. It will diffuse into the cell. While potassium, on the other hand, we just built up this massive potassium gradient in the cell. It will want to go out of the cell. So let's build this gradient. Let's build our sodium-potassium gradient. And to do that, we're going to move on to our next layer. So using green as our example, we're going to have a conformation change right now. So we're going to have some sodiums inside the cell. And we're also going to have some potassiums outside the cell. So now we're talking about this one, so we can get rid of our top layer. Just to help with the confusion a little. So we have our potassiums that we need to build the gradient inside the cell with. And we have our potassiums that we want to push out of the cell so we can build our gradient. Our conformation change is going to look like this. So in a resting state, our sodium-potassium pump will be closed. We'll have a closed example. And what we'll do is we'll load up some sodiums that we want outside the cell. So we'll have 3 sodiums inside the cell. We'll push them out. So we'll have 3 sodiums. So we'll load it up with 3 sodiums inside our sodium-potassium pump protein. However, it just doesn't spontaneously change. We need some sort of energy. So what we're going to do is we're going to attach an ATP phosphate to it. So we're going to take a molecule of ATP, convert it to ADP. ATP is adenosine triphosphate versus adenosine diphosphate. So we used to have 3, now we have 2. What happened to that extra phosphate? We're going to stick it onto that pump. So the sodium-potassium pump becomes phosphorylated. And that phosphorylation is going to come from ATP. What this phosphorylation does is we're going to do a conformation change. So we'll move down to our next figure. Notice how in this example it was facing inside. We're dealing with stuff inside the cell. Once we attach that phosphate to it, we're going to have a conformation change, and now the pump is exposed to the extracellular side. So let's see. I'm going to get rid of this ATP process because we just saw that it was phosphorylated. And maybe I'll draw ATP just so we can remember. But, so we just did a conformation change, and our sodium-potassium pump is now facing the extracellular environment. So what happens here? Well, we have, like I said, we still have our potassium outside the cell, but we still have these 3 sodiums that are inside the pump. So we're going to pump out our sodium. So these will leave. So now we have 3 sodiums that leave the inside of the cell. So now they're in the extracellular environment. So let's draw that in. So now we have 3 extracellular sodiums. What the body is going to do then is we're going to load some potassiums in the pump. So now we're going to have 2 potassiums. Notice we exchanged 3 sodiums now for 2 potassiums. So it's a 3-to-2 ratio. 3 sodiums for 2 potassiums. All right, so now we have 2 potassiums inside this channel. So 2 potassiums inside the channel. What do we do? Well, we do know that it's still phosphorylated. Our cells are going to use the ATP that's inside the cell to phosphorylate it here, and we still have this phosphate attached. And attaching the phosphate is going to cause the conformation change, so now we're exposed to the extracellular, our sodium leaves, we're going to gain 2 potassium, and our potassium are sitting there. When we remove this phosphate group, when we dephosphorylate the sodium-potassium pump, so if we cleave that bond, phosphates are just energy bonds. So if we remove the energy from that bond, when we cleave off the phosphate, our phosphate will leave, and what will happen? We'll have a free phosphate, and we'll have a conformation change back again. So notice, originally we were exposed to the internal environment, we then changed it to the outside environment, now we're back in. So we had 2 potassiums still in that channel, and we now have 3 sodiums outside the channel. And now what will happen is these will diffuse out into the cell. Okay, so those diffused into the cell. So what happened? Well, we exchanged 3 sodiums, which are now in the outside, with 2 potassiums, which came into the cell. We're creating our sodium and potassium gradient, and that gradient is used for depolarization. So what this pump is effectively, it's creating a gradient for depolarization. And then when the cell depolarizes, this sodium will go down its concentration gradient. There will be so many sodiums outside the cell here. And I'm going to get rid of the rest of this. There we go. And get rid of all this. So now we created all these sodiums outside the cell. We have all these potassiums inside the cell. When the cell depolarizes, we're going to have channels that these can go down the gradient into. All potassium will leave. So what the sodium-potassium pump does, we're creating a sodium and potassium gradient. We're going to exchange 3 sodiums for every 2 potassiums. We're going to use 1 ATP, which will cause that conformation change in the protein, thus creating a gradient. If you found this video useful, please like, please comment, please subscribe. If you have any questions, feel free to ask. Otherwise, thank you very much.